Totally new, totally different. Good afternoon. It works. All right, so what are we talking about today? So we are in the midst of module 2, which is all about the dynamics. So module 1 was all about DNA and sort of how to annotate regions in the DNA. Module 2 is all about sort of bringing DNA to life. So the transcriptome, the epigenome, the folding, all of that is sort of active things inside the cell. And we're going to do quite ambitious kind of journey. We're going to try to cover the last part of the last lecture and all of this lecture, which is, you know, very ambitious. So just to recap, we talked about gene expression analysis. We talked about how there's 2 different technologies. One is microarray technology of hybridization. The other one is RNA sequencing. And we basically said that you could count reads and then map them to the genome. So today we're going to be talking a lot more about that. Then how the resulting data matrix effectively looks like a bunch of columns, each column capturing 20,000 genes in a given condition, and then enabling us to look at the expression pattern of a gene across conditions given the right set of normalizations. And then we can start asking about gene similarity questions and experiment similarity questions. We saw how we can do unsupervised learning with clustering. First by k-means, where you have a set of centers which you keep pushing to the right middle of the points assigned to them, and a bunch of points that you keep assigning to the nearest center. And then we saw the fuzzy k-means version, where every point is probabilistically assigned to all of the centers, with a probability scaled by the probability, with a likelihood that that point was generated, was sampled from that probability distribution. And then we saw how that algorithm was in fact the expectation maximization optimal way of updating this Gaussian mixture model that basically had these univariants and these points sampled from that univariance. And then we saw how k-means and fuzzy k-means in fact had both an algorithmic and a probabilistic formulation that sort of matched. Then we saw how EM was in fact so much more powerful than just k-means, that it would allow us to look at different priors, different shapes of these distributions, different spread of these points by adjusting these variants in the covariance matrix, and also different ways of selecting the update and the assignment, either using the full density or by sampling the posterior or by choosing the closest point. Is everybody with me so far? You guys remember all that? Great. And then we saw hierarchical clustering and how, in fact, instead of choosing clusters, we could just build a nested grouping of all the clusters and then choose a number of points by just simply cutting things off. And how we can evaluate the goodness of a cluster using this hypergeometric distribution to basically tell us how likely am I to obtain that many orange points in a random set of blue selection, given the overall abundance of these orange points. Okay. And then we got into classification. So the second half of this, which is basically saying, well, we can do generative classification or discriminative classification. And the idea is that based on your samples, you infer from the known labels a probability distribution. And that's easier than what we were doing in the unsupervised case, because it basically says, well, we have actually labels for these points. So we can fit either a parametric model with a mean and a standard deviation and some priors, or a non-parametric model by learning some kind of binning approach, okay? And we can basically obtain this probability of obtaining, of generating any point from a given class. These are the class conditional generation probabilities. And then how we can combine, you know, so the next part that we didn't talk about too much is how do we combine multiple features, okay? And then the idea here is that you may have multiple lines of evidence that pertain to your class of interest. For example, if you're trying to classify mitochondria proteins, so proteins that are transcribed, whose genes are transcribed from the nuclear genome and then are transported over to the mitochondria. So the mitochondrion is this thing that kind of co-lives with us. It's basically an ancient species that was engulfed by a pro-eukaryote. So, you know, all eukaryotes today have organelles within them. Plants, for example, have chloroplasts, and we and plants have mitochondria, which are basically the energy producing machinery inside our cells. These mitochondria used to be a separate species, which had its own genome, and it was engulfed and specialized to carry out only energy production. So that mitochondrion still has its own genome, but it shed a lot of the genes that could have been, that can simply be produced by the nuclear genome. So the mitochondrion mostly has 13 genes nowadays, which are all part of this energy chain. And then there's another 600 or so proteins that are transcribed from the main genome. How do you find all of the mitochondrial proteins from the genome? Well, you could look for targeting signals. These are signals at the RNA level and also at the protein level that basically tell a transcript or a protein product, go to the mitochondrion. How? By basically recruiting specialized proteins that then sort of move along the cell, okay? Or you could look for protein domains that are much more common in mitochondrial proteins, or the co-expression patterns, because all of these genes are co-regulated very often and therefore are more likely to be co-expressed. Or you could look at the mass spectrometry evidence from mitochondria isolation that basically can say, well, how many spectra do I see, how many proteins do I see that sort of come from that part of the cell. Or you could look at homology, like evolutionary relatedness with other proteins, and so on and so forth. Okay, is everybody with me so far? So you can use these multiple lines of evidence and you can say, well, given all of that evidence together, how can I classify something as mitochondrial? Therefore, you can look at the probability that given all this evidence, you're class 1, or the probability that given all that evidence, you're class 2. And if you multiply that by the prior, you basically get effectively that ratio, because of the simplification of P of feature, allows you to basically calculate the probability of the class given the data. But the problem is that we can easily compute the P of data given class for each of the classes individually. Computing that joint probability is very hard. Everybody with me so far? So to do that, we basically, in naive Bayes, make a very simple assumption. We basically say that the joint is just the product of the margins. It's a huge simplification, but it basically allows us to argue about each of the features independently. So what this is a fancy name for is making an independence assumption. And maybe it's not that fancy of a name since it's naive Bayes. It's at least giving justice to how naive it kind of is. But it basically says, well, I'm going to naively assume that the classes are independent of each other, that the different features are independent of each other. And as we mentioned in the last lecture, it's not always real. It's not always true, but it's often, you know, giving you a good answer, okay? There's two types of models, the ones that are wrong and don't work, and the ones that are wrong and work. There's no class of models that is actually right, because the world is very complex. So you're never going to get the world right, but what you can do is actually, at the performance level, achieve something that is, you know, performing well. So therefore, you can estimate the individual distributions of each feature and just multiply them together. Sounds good? So that's effectively what naive Bayes discrimination does. It basically says, my generative function is now the joint of all of these, which is the same thing as the marginals just multiplied with each other, which is simply this discriminant function evaluated for each of the features and then added together in log space. Okay, so it's effectively an additive score across these many features. Everybody with me here? So we can just simply compute this as the sum of log scores. And then once you have predicted some mitochondrial proteins and you've predicted some non-mitochondrial proteins, you can basically ask, how are my predictions matching true annotations that comes from some other medium? And that gives you true positives. That gives you true negatives when you're doing well and they agree. And ideally, most of your samples are here. But every now and then, you're going to be predicting something as mitochondrial and it won't be, or you're going to predict something as non-mitochondrial when in fact it is. So we're going to be talking about sensitivity, which is the fraction of all class 1 proteins that are mitochondrial that we correctly predicted as mitochondrial. And the specificity, which is the fraction of non-mitochondrial that you predicted as non-mitochondrial. Okay, so, and you need both. You can't, I mean, it's easy to have awesome sensitivity, right? You just predict everything as mitochondrial. It's easy to have awesome specificity. no false positives. You know, you can just predict everything is non-mitochondrial, okay? But the problem is it's hard to do both, right? So that is actually sort of where the money is, basically. So you're trying to maximize both, and therefore your error can be quantified as sort of these off-diagonal quadrants. And you know, there's many ways of doing that. So in practice, you basically take all of these different domains together, and then you can learn these class conditional probabilities. You can infer them by binning, and then you can use them to predict, and then you can combine the evidence together, and then see that in fact these naive Bayes approach outperforms, you know, many other approaches by being able to combine a lot of evidence together. I mean, it's not amazingly different, but it is higher sensitivity and higher specificity than any other one. So basically, the further up this diagonal you are, the you know, the better the method is. Okay. So this is all generative models. This is basically saying I'm going to try to use the entire probability distribution, model it, and then you know, use something based on that. What support vector machines do and what discriminative methods do is say, I don't care to model everything. I'm only going to model the things that are the most relevant for my discriminative choice. And what support vector machines in particular do is they select a set of points that are the closest to that discrimination boundary, and they use them as effectively support vectors onto which you can basically guide a discriminative function, a threshold between them, okay? And the question is, of course, what line should we choose? And what SVMs do is that they basically say, well, we're going to choose the line that maximizes the distance from that midpoint, from that separation, okay? So in 2 dimensions, it's very easy to visualize. It's basically whatever line goes through that plane that allows me to maximally, you know, discriminate these points. Raise your hands if you're with me. Awesome. So, and then your support vectors are effectively the points that support that line, okay? And then the formulation is in linear algebra very simple. It's like the dot product between x and this vertical line w that's perpendicular to your separator line. That minus that some, you know, threshold has to be greater than 1 for the points that are on this side, and then, you know, less than -1 for the points that are on that side. And then in the middle, we just don't classify things, okay? So we're basically looking for the separator with the largest margin, and that's how SVMs work. It's, you know, very straightforward, at least in this very simple two-dimensional plot. Where SVMs get really powerful is because they're not just looking at 2 dimensions, they're looking at many dimensions, and they're not just looking at linear functions, they can have non-linear discriminative functions, boundaries. How is that possible? By basically mapping the space into a different function, into a different space. So for example, if I'm trying to map a function that distinguishes the green points from the blue points here, I can't do it in this particular space. However, I can look at the 2-dimensional space between that includes both x and x squared. So if I compute x squared, I can then find a very simple discriminative line that basically separates the blue points from the green points. Okay, who's with me here so far? Awesome. So the key and the power of SVMs really comes from the fact that by being able to transform my data and look at additional dimensions beyond just the primary data, I can effectively find discriminative functions that can be very complex, okay? And then there's a whole family of functions that preserves the, you know, a lot of very cool, interesting properties. Namely, because SVMs work on that dot product with this vector, if you basically select functions that have, you know, this dot product preserved, you can effectively find functions that will be very easy to map, very simple to map, very, you know, easy to compute on, and yet can have very complex properties. So instead of worrying about just the x, I'm going to worry about the dot product of these kernels, and then I'm going to of these functions, and then I'm going to define a kernel function that basically deals directly with the dot product of these functions. Okay, and that allows me to now generate all kinds of interesting bases that sort of capture that. Okay, so instead of computing this on individual points, I can compute this on pairs of points, and then not just work with the dot product of these points, but actually work with much more interesting functions of these points. So I can still compute my SVM onto those. Yes?
What is the kernel?
The kernel is basically the function that you use on, so it's a mapping function that basically starts from pairs of points and then maps them to some space.
Okay, that's
And
the higher dimensionality.
exactly, that's where the higher dimensionality comes from. So you can basically think of the higher dimensionality as this f function, or for pairs of points as this kernel function that basically takes in pairs of points and then computes that higher dimensional object. So that allows you to basically do discrimination in much more effective ways. Okay, and there, you know, there's a lot of examples of such functions: linear functions, polynomial functions, radial basis functions, sigmoid functions, all have these non-linearities that allow you to sort of carry out discrimination in much more interesting and powerful ways. And then SVMs have been applied to expression analysis in practice very early on, very powerfully. So for example, you know, this group at the Broad Institute, or at least the precursor of the Broad Institute, had 38 bone marrow samples, and they didn't know if it was ALL or AML to different types of leukemia. And what they found is that based on this microarray measurement of 6,000 genes, they could basically build an SVM that allowed them to discriminate things very strongly. And the support vectors ended up being genes that were, you know, quite meaningful in cancer. And, you know, this was, you know, one of the early sort of AI lab / Broad collaborations. So there's other approaches where you can basically bring both clustering and classification together. So you can basically have only some of the points being labeled, and that gives you the classification part, but many more points available, which allows you to learn the shape of the space, of the distribution of that space. And you could basically look for, if you only had the label points, you would say, okay, great, I have a great discriminant function. But if you have the full distribution, you would say, oh, this is you know, a much more interesting way of clustering the data and separating the data. And having both together allows you to basically both learn the structure of the point distribution, the overall point distribution, by using all the points, and the classification task by using the subset of points that are labeled. Okay? Raise your hands if you're with me. Awesome, great. So this was lecture 6, and here's lecture 7. Okay, so basically lecture 6 was all about gene expression analysis, k-means, hierarchical clustering, naive Bayes classification, and then support vector machines. Okay, and sort of this discriminative versus this generative classification. So 2 ways of doing clustering and 2 ways of doing classification. Okay, one partitioning, one agglomerative, and then one generative and one discriminative. Okay. So today we're continuing the journey on understanding gene expression analysis and understanding RNA. Okay, and the first thing that we're going to do is glorify RNA beyond just simply this transfer molecule, and actually understand the complexity of RNA functions. Then we're going to look at that very first slide of yesterday's and today's lecture, the first data slide, that basically tells you, oh, you can have this microarray in the middle, or you can do things de novo by sequencing. And we're actually going to look at ways of reconstructing transcripts de novo. So rather than just mapping things to known intervals and then counting the number of reads that map to those intervals, how do I find splicing patterns of an RNA? And how do I reconstruct the genome de novo with or without a reference genome? Because again, I'm doing sequencing. And then the last thing is going to be building on this part here to basically look at how RNA itself can carry out functions. So we're going to look at the folding of RNA in 2 dimensions and by extension in 3 dimensions. Sounds good? So I showed you this slide in the first lecture, basically saying that pretty much every living thing on Earth today can be traced to some kind of common ancestor. Okay? And here I'm showing you only eukaryotes, and even yeast has mitochondria, so it is still one of the species that has engulfed eukaryotes. But the tree of life is much more complex. And before eukaryotes were around, and before bacteria were around, and before archaea were around, there was something there that was the common ancestor of everything. And the question is, what did that something look like? And that something probably had some combination of RNA, DNA, and protein. But the question is, what came before that? And the answer to that is that when you look at the central dogma of biology, you basically realize that RNA, protein, and DNA are all intertwined. They're all sort of generating each other and transcribing each other, reverse transcribing each other, etc. But there's one element in particular, RNA, which plays every conceivable function. So RNA is basically the messenger, which is the way that we think about it in very simple terms in gene expression analysis. It's mRNA expression. But RNA is also the molecule that translates itself into protein with transfer RNA. It is also the molecule that carries out the translation with ribosomal RNA, which then makes the ribosome and then brings in tRNAs to translate RNA into protein. It is also carrying out dozens of other functions, some of which are very ancient, some of which are very modern. So if you look at the central dogma of biology that I showed you in the first lecture, there's this very easy way of picturing life, which is that DNA makes RNA makes protein. Information transfer, that's the code, that's the messenger, and that's the catalysis or the function. And DNA is today specialized to do information storage and cross-generation transmission. RNA today can be thought of as specialized as a temporary copy, which is very boring. And protein is, wow, the interesting thing that carries out all cellular functions. But it turns out that RNA can also store genetic information in the same binary form. RNA can also transmit information, and RNA can also carry out enzymatic reactions. So RNA is involved in almost every process at the core of a cell. I mentioned messenger RNA. I mentioned the ribosome with ribosomal RNA. I mentioned tRNA, transfer RNA, that basically transfers information between RNA domain and protein domain. Why? Because it has two ways of acting. The first way is by complementarity, and that's where it recognizes codons. The second way is because of its three-dimensional fold, where it can actually mimic anything that a protein can do. So it can basically have one front, one side of it acting as a DNA, and one side of it acting as a protein. And it can catalyze these reactions while at the same time reading DNA information. But these are very simple, and again, the existence of a tRNA molecule, of the tRNA as the transfer way between messenger RNA and protein, was in fact hypothesized by Francis Crick 10 years before it was actually recognized to be so, by sort of thinking about it clearly and saying, this transfer molecule must have the, must be made of RNA. Yeah. Every DNA, every RNA that's single-stranded DNA. RNA has a U instead of a T, and that makes it much more foldable. The T base does not make DNA as easily foldable. So single-stranded DNA, yes, can actually, basically, it will not want to live in isolation. Single-stranded DNA will actually fold up. But because of this difference in molecules in the bases, it actually has a much more rigid folding pattern. But if you have single-stranded DNA inside the cell, it will very often sort of, loop up like that. So it'll have this like unbound part, and then it will bind there. But the problem is that you need topoisomerase. You basically need to uncoil it, just like the string on your, on your bike lock. Kids a little older than you guys remember phones. You probably have never seen a cord of a phone. But the concept is that if you uncoil it, it'll just like, spring back in. And the same with your bicycle lock. If you uncoil it, it'll have all this tension. And basically, in order to sort of fold it onto itself, or your watering hose, if you loop it, you have to actually do a turn. And same thing with sailing. Like when you take a rope and you fold it, you need to actually constantly do this looping. If you take a single strand of DNA, it'll fold up, but it won't have the ability to sort of loop together because it'll have all this extra coiling. And that's where topoisomerase comes in and it cuts it, and it allows it to loop around and then, rejoin it. So yes, DNA can do that, but it's very often found in very long molecules that don't have as much flexibility to do that. But basically, if you leave a self-complementary piece of DNA by itself, it'll actually sort of fold up and then rotate around. and then you can just, re-uncoil it. But that's a great question. But again, as I'm going to argue shortly, DNA was invented at a time when RNA already had all the cool functions. So it didn't have an evolutionary niche to capture any of these functions because RNA was doing the job fine. Thank you. All right, so RNA is actually involved in regulation with double-stranded RNAs, antisense RNAs, microRNAs, in degradation, in replication with siRNAs, microRNAs. It has all kinds of functions in the processing of transcripts with the 6S RNAs and exis RNA and rRNA. All of these guys are actually long non-coding RNAs that help code the genome and silence chromosomes. In processing of the transcripts with SNO-RNAs and small nucleolar RNAs and self-splicing introns, etc. And it's basically extremely diverse in the kinds of functions that it can carry out. And if you look at this image of a nucleus and the cell outside, you basically see that it is involved in translation, in transcription, in splicing, in modifications, in post-transcriptional repression, in translation, and so forth. So it plays this huge number of functions. But the much more interesting part, the much more exciting part is not how diverse it is today. It is the fact that if you look at the core of many of these machineries, it seems that the ancient form was RNA, not protein. The ribosome has both proteins and RNA together. And when the structure of the ribosome was formed, it was realized that at the core of the ribosome lies only RNA, that the entire catalytic process is carried out only by RNA, that protein only serves stabilizing roles surrounding that core. And this is incredible because this is, perhaps the most complex machinery that has ever been invented. And it seems to be extremely ancient, that basically, it has not been optimized further by new proteins coming along. Everybody familiar with the basic function of the ribosome? So basically the ribosome takes a tRNA, which is this transfer RNA, that basically serves as an adapter between a codon, like AGT, and the corresponding amino acid, okay? And it basically fits two different tRNAs inside it, and it basically just moves like a crank. It basically, moves around, bringing in the new tRNA and then kicking it out, and then bringing in the next one, and then, it's kind of like gears, moving aside. So that's where the small subunit and the large subunit work together to basically effectively carry out translation. Sounds good? So, and then again, they're at the core of the catalytic production of spliced messenger RNA. RNA can actually replicate itself. So RNA can basically serve both as a self-replicant and also as a general replicant of any kind of RNA template, which is quite remarkable. And RNA can mediate enzymatic reactions. So it can basically do the things that we used to think are unique to proteins. And here's some enzymatic reactions, for example, self-splicing introns or processing of tRNAs. It can also serve as a sensor of the environment. You can basically have these ribose switches. These are RNA-based loops that will sense something in the environment, change structure, and then enable the catalysis of something else, or enable even translation of their own protein. So the message that I'm trying to give you is that RNA could get by without DNA and without proteins to basically carry out all of life. Every single form of self-replication, self-modification, folding, and all of that could be, even today, carried out by RNA. And in fact, that's exactly how it was, according to the most likely models, before DNA and proteins were even invented. There used to be something that we now call RNA world. At the time, I don't remember how they were calling themselves, but language was probably not as complex. And basically, the concept is that in this very pre-life era of our planet, RNA molecules self-assembled and sort of were generated by probably hydrothermal vents, not the black smokers, but the white smokers, if you wish, with molecules that can be created by natural processes, just simply chemical and physical processes, that will then form polymers by being stacked on top of each other into these sort of small shaped tubes and holes that are more or less the size of a cell today. And this kind of compartmentalization allowed life to eventually emerge as this RNA life. And then you basically start with these RNA molecules that are sort of self-assembling, and somehow you have to kickstart the process of life. And that means self-replication, okay? So the very short summary, and this is exactly, you know, not written in stone, it's stuff that we have inferred from a lot of evidence. The idea is that from this RNA world, we got to today's world by RNA starting to evolve a little more efficiently. You basically have initially these clay minerals that guide single-stranded RNA formation. These detach and fold and catalyze reactions. And then the kickstart of life comes from self-replication or cross-replication, where one molecule favors the production of another molecule and vice versa. And the moment you close that loop, you have life. You basically can have the process of evolution starting. You can have improvements that self-propagate. Who's with me so far? Awesome. Who thinks this is kind of cool? Yay, awesome. And then RNA can start making more RNA, and then the process of life begins, the process of metabolism and compartmentalization and sort of the sense of self versus non-self, and so on and so forth. And then you basically have this RNA world that was living much more complexly than any kind of pre-life form, with replication, with sensing, with catalysis, with compartmentalization, and so on and so forth. So RNA then starts evolving more efficiently. You have mobile elements and viruses and, you know, the process of evolution. Then you start having splicing, where you can process these very long, cumbersome molecules into different types of introns. So group 1, group 2, and group 3 introns are all RNA-based, so these can form around. And then you have general splicing. So the spliceosome, which is mostly RNA, you know, is evolving as a machinery that can splice any kind of RNA molecule. And then RNA basically is tired of doing all the work, and it invents robots, known as proteins. And that's where the ribosome, the incredible machinery that's still in every living organism today, is invented. And this is all RNA at the core. And the ribosome allows you to make protein. So you now have RNA and protein that can now work together. And that's when RNA is tired of doing all the replication. So it just says, okay, let's make a form of me that's a little more stable. And that's when it makes DNA, okay? And reverse transcription happens in a protein-based form, which is kind of cool because by then you have proteins. And then from DNA to RNA, again, transcription, well, you already have proteins that you invented to make DNA. So you can use a protein which is much more effective and efficient because it has 20 building blocks rather than just 4. And then RNA, protein, and DNA start specializing, and then DNA effectively becomes relegated to just information storage. It's kind of like the boring molecule. Proteins do catalysis, sensing, structural roles, and so on and so forth. And then RNA still does pretty much everything. Okay, sounds good? Who used to think that DNA was the coolest molecule? All right, who now thinks that RNA is the coolest molecule? Awesome. Great. So this is basically why RNA is so darn cool, because, you know, all our ancestors were RNA-based, it turns out, and because even still today, inside the cell, RNA carries out a myriad of different reactions and functions that we're still only now uncovering. Okay. Who feels that they've learned something? Yay. Awesome. So we talked about the functional diversity of RNA molecules. We're talking about ancient housekeeping non-coding RNAs and the RNA world hypothesis. We talked about young non-coding RNAs as additional regulatory layers. So microRNAs were recently invented, long non-coding RNAs were recently invented, and so on and so forth. So RNA is still very active and still taking on new roles. So RNA is very important, very cool. So now we're going to talk about two things with RNA. First, how do we go from RNA sequencing reads to transcripts, and then how do we actually fold RNA in 2 dimensions and ultimately 3 dimensions, okay? So before we do that, we're gonna take a 1-minute break. Stand up and stretch. You're doing it right. All righty. So remember this very first slide. I said, oh, we basically built these probes for measuring microarrays, or we can count RNA molecules by simply looking at how many reads map into each of these exits. Well, this part is much more complicated. So basically, the first part is generating the experimental data. So you basically start with some mRNA or some total RNA, and then you remove contaminant DNA, you remove these double-stranded molecules, and then you fragment it, you reverse transcribe it into cDNA, that's complementary DNA, and then you basically ligate specific adapters, and you carry out PCR amplification by starting from these adapters. You can select a range of sizes, and then you sequence the ends of these molecules. So you can basically sequence from both sides of the cDNA. So you basically can have pairs of reads that are near each other with some distance because you selected that range of distances. Okay? Raise your hands if you're with me on this part. Great. You then want to reconstruct the transcript of every gene in the genome, also known as the transcriptome, from these RNA-seq reads. So what you can do is actually align these reads to the genome and then figure out where they map and assemble transcripts from these spliced read alignments. So you basically have this read that splices continuously, and you have this other read that maps continuously, and this other read that maps in a splice form. Why? Because messenger RNA is actually spliced, because introns are excluded, they're folded onto themselves and then cut out. So you end up with a single RNA molecule that seems contiguous, but when you map it to the genome, it actually maps to different parts. Is everybody with me on this part? Great. So you can basically assemble these transcripts from spliced alignments. So that's one approach. You basically map everything to the genome first. The other approach is you map these RNAs to each other. So you assemble them on top of each other. You basically say, oh, this purple one matches that purple one and that purple one. So I'm going to construct a single molecule. I can infer a single molecule that existed, which actually corresponds to a single long transcript. And there's another molecule, and these might map to the same region of the genome, but with different splice forms. Everybody with me? So let's now look at more detail into how these map to the genome methods work and how these Align-de-novo methods work, okay? The map to the genome versions basically started out with bowtie, this very fast short read alignment that uses the Burrows-Wheeler transform, and there's a typo here. And then once you have these, so the first name was bowtie because it's got BWT, Burrows-Wheeler transform. And then the Tuxedo seat was born. The second one was basically Top Hat, that was basically working on top of bowtie, and it was sort of linking things together. And then Cufflinks with transcript reconstruction, and then Cummerbund with additional applications and so on and so forth. So and that basically became the Tuxedo suite. And there's a lot of players of that Tuxedo suite. One of them is Loyal Goff, who was a postdoc with me and with John Wren. He's now a professor over at Johns Hopkins. and he developed cummerbund, but basically there's a big family of happy people all working together. I was having a call this morning with this guy at 9 a.m. I was chatting with Steven Salzberg, who's a very cool guy. Anyway, so how does this suite, for example, carry out step one? Well, step one is you want to map reads to the genome. And what do you want to do first? First, you want to look for unspliced reads because they're easy. So you first look for all of these reads that you can map to the genome contiguously. And then you basically collect all of the reads that were not initially mapped. And that's where you basically map together the non-contiguous reads. And this mapping, we're going to talk a lot more on Tuesday when we go in detail through the Burrows-Wheeler transform operation for this mapping. But it's similar to BLAST. It's basically got some kind of hashing approach, only it rearranges the genome beforehand to make this mapping much more memory efficient. Okay, and I'm going to talk a lot more about that. Okay, so on the Tuesday lecture in genomics, we'll actually see how the Burrows-Wheeler transform actually works in detail. But once you have mapped these reads contiguously, you want to deal with the splice reads. So you basically have mapped those, and now you say, okay, well, here's this read here that I know maps contiguously, and its pair has not been mapped to the genome in my contiguous step. So what I'm going to look for is bases at the beginning of it that map somewhere, and bases at the end of it that map somewhere nearby. And that's a much easier search because I already have a seed from the first one that tells me roughly where it goes. So I can basically search for splice sites that interrupt that particular read. Raise your hands if you're with me on this one. Awesome, great. So what TopHat basically does is that it aligns these read fragments, it breaks them up into little pieces, and then it aligns them, and then it looks for GTAG pairs that are sort of consistent with a splice site interrupting these reads. Okay? And once you have these splice sites, you basically can sort of form longer transcripts. And then the last step is you'd like to actually do the quantification of how much expression did I have from different transcripts that all map at the same location. And the idea is the following. You basically have two different transcripts, one short and one long, that both map to the same location. You basically, let me just back up just 1 second. You have here two different splice forms that are mapping to the same region of the genome. And you'd like to know, well, if I have 500 reads, how do I assign them between this transcript and that transcript? Well, the simplest idea is if this exon, for example, is unique, then I can map the overall expression level of this transcript versus that transcript, and then infer the proportions that I see here and apply these proportions to the overlapping exons. So I can basically take, the non-overlapping portion. So here, for example, I can tell where this one is how much this one is transcribed, infer from that how much I would expect elsewhere, and then from this section here, for example, how much this is transcribed, and then extrapolate to partition the rest of the reads accordingly. Who's with me so far? Awesome. But the problem is that you need to correct for the total gene length. Why? Because the longer the transcript, the more reads will map there. The more opportunities you have of catching an RNA molecule there. So you have to adjust for the total gene length. You also have to guess what splice variant each one is coming from. And there are probabilistic models that have been developed in order to do that, where you basically have some table of compatibility that basically tells you how consistent are my different fragments with particular transcripts, and I can partition those accordingly. And of course, with long read sequencing technology nowadays, some of these steps are becoming less and less central. And the problem, of course, is that as you map more reads, your estimate of the expression level of every gene gets adjusted. So there, you can actually do that iteratively. You can basically have an expectation maximization approach that finds the most likely assignment of reads to transcripts, and that's sort of at the core of many of these methods. Okay. And then the last step is that there are actually systematic biases in reverse transcription that favor particular combinations of nucleotides and k-mers that you can adjust for, and based on that, some RNA molecules will have many more reverse transcripts being generated because reverse transcription works more efficiently with their sequences. So you need to correct for that to figure out how many RNA molecules were there actually in the cell. Is everybody with me here? Awesome. All right, so you do all that, and in the end, you basically end up with transcript map read counts, which can then be normalized both for length and for the total depth of sequencing. Why? Because the more you sequence, the more reads you will have for everything. So that's the M part, basically per million fragments of sequencing. And then the normalized for length, where longer RNA transcripts have more reads mapping to them. That's the PK part. So basically what you're counting is the number of RNA-seq fragments per kilobase of transcript length per total millions of RNA fragments that are mapped, okay? So the more you sequence, the more you normalize, and the longer your transcript, the more you normalize, and then you count fragments ultimately. Who's with me so far? Awesome. Great. So what we talked about is the first part of transcript reconstruction. How do we map to the genome? And as an example, we use this Tuxedo suite, and specifically Cufflinks, where you first map individual reads, then spliced reads, and then you quantify transcript abundance. The other approaches are aligning the transcriptome de novo, and one example of that is the Trinity approach, also developed here at the Burrows. So this actually does not require a genome, and this is important because you can actually study a lot of non-model organisms. So, you know, you can figure out the expressed gene content, the transcript abundance, differential expression without ever having a reference genome. And the idea for that is that you're going to basically create de novo assemblies of these transcripts. You basically are going to assemble these fragments and then figure out bifurcation points where presumably different splice variants are living, and so on and so forth. Okay, so that's, for example, you know, two different copies of a gene that have slight variation, or this could be the two different chromosomes of a heterozygous individual that basically has sequence differences between them, or this might actually have sequencing errors, and the other one doesn't. So the first thing you do is that you break up all your reads into small fragments, and then you assemble them into longer chunks. And then you build together a graph that tells you how these are related to each other. And then once you have this graph, you can collapse it into one node here, another node here, another node here, another node here. So basically, you don't need to have these individual k-mers anymore. You can basically collapse them into longer segments. The basic idea here was developed by Pavel Pevsner back in the early 2000s. And the idea of that is a De Bruijn graph for sequence assembly. The idea is that I'm going to basically partition everything into tiny little chunks and then put arrows between sequences that differ by one nucleotide that are this, you know, k-1 overlapping k-mers. Okay, and then this gives you a directed graph that basically tells you how to traverse every k-mer into every other k-mer. And then that's sort of the basis from which you then construct these longer assembly graphs, okay? And then you collapse this De Bruijn graph, and then you end up with something that captures your whole transcriptome, but still has some ambiguity. For example, you don't know if this exon goes with that exon or with this exon. For example, I could take the top path here and the top path there, or maybe the top path goes with the bottom path. And that's sometimes very hard to infer. And that's sort of where you have to do some graph reasoning, okay? So then the idea is, how many ways do I have of traversing this graph? I can have the blue way, the red way, the green way, the orange way, and each of them corresponds to a different complete transcript, okay? This is still an unsolved problem. Basically, I don't think that any method can fully capture the true splice graph and the, you know, the corresponding transcripts with great accuracy. So long read sequencing is one approach. Probabilistic assignments is another approach. But the, you know, the short read aligners are sort of really not fully resolving it. And part of the reason is that biology is also complex. I mean, an RNA molecule might actually make a splicing decision here quite independently from a splicing decision there. Sometimes, of course, there are signals in the RNA that will guide the splicing machinery to make consistent decisions along different phases. Other times, it's just combinatorial, and you actually have all, splice forms. So, resolving this computationally, we might simply not have enough signal, just like the cell might not have enough signal for most transcripts. All right, so these are the two approaches. The first one is you map to the genome, first the individual reads and the spliced reads, and you quantify the transcripts. And the other one is that you start building these linear contigs and you assemble them together into an assembly graph, which you then resolve into individual transcripts. Is everybody with me so far? Awesome. All right, so the last step is we want to calculate differential expression using those quantifications. And to do this, you need to deal with a lot of the technical, biological, and simply, uncertainty-based variability. And that's what tools like CuffDiff do. They basically seek for these systematic differences by building models of transcripts in one condition, transcripts in another condition, and then maintaining a lot of that uncertainty when resolving the individual transcripts. And you can basically visualize these using, for example, volcano plots. These are something that you'll very frequently use in your final project. This tells you both a log fold change and the statistical significance of that change. Because something might be very significant, but not a big fold change, and other things might be a big fold change, but not significant. But most of the time, you see this correlation between the two. And then there's also, average expression versus fold change, where you basically need to have both in order to have reliable estimates. So your significant ones are the red ones in this particular case. And it's very hard to tell significant changes when the overall expression is very low, but it becomes much easier to detect subtle changes of like 1.2-fold change for something that is actually quite abundant. Okay, so this is how, so basically the key points here are that RNA-seq is an extremely versatile method for transcriptome analysis, enabling both quantification and discovery of novel transcripts. And then both genome-based and genome-free methods exist for transcript reconstruction. Expression quantification is based on both sampling and counting reads derived from the transcripts. And then fold changes that are based on very few reads lack statistical significance. And then there's many analysis frameworks for supporting biological investigation. Okay? Who's with me so far? Awesome. Great. So we're going to do something unusual. You guys can have another 30-second break. So stand up again. We're going to stretch again. And then now we're going to switch to the RNA folder. Please, everybody, actually stand up. Who's feeling grateful that their lecture is being recorded? Who feels that they'll never have time to go back to it? A lot of my students are saying, you're taking pictures all the time. Do you ever go back? I'm like, yes, I do. Between 3 a.m. and 5 a.m. All right, let's get back. So as I mentioned earlier, RNA is an incredibly versatile molecule. It can carry out pretty much all of the functions of life by itself. And it still does in many species. And there's many viruses that are entirely RNA-based. There's a lot of life forms that are entirely RNA-based. So how does it do that? By folding onto itself and creating these complex three-dimensional shapes. So what we're going to talk about now in the last third of the lecture is folding of RNA and how we can actually predict the folds of RNA because they lie at the basis of all the functions. So we're first going to introduce the representations for RNA folding, then we're going to look at biophysical energy constraints, and then parsing-based approaches, and then phylogenetic approaches, and more advanced approaches. Okay. And the lecture goes until about 5 p.m., so you guys have nothing else scheduled today. So this is the primary structure of RNA. It's basically a single-stranded molecule. And the secondary structure is effectively telling you how the single strand folds onto itself. So you can see here that the same strand loops around and then forms base pairs with itself. And again, base pairs here with itself, base pairs here with itself, okay? And it's based on that secondary structure that the RNA then folds into these complex protein folds that allow it to have, complex three-dimensional folds that allow it to have a diversity of functions as complex as that of proteins. You can basically see here a structure of the tRNA, tiny little molecule that sort of folds onto itself in these ways. And you can see here the base pairs that are formed. So you can have, UA, CG, UA, and then here, CG, AU, and CG, AU, CG, AU, CG, AU, GC, etc. Okay, so there's a lot of sort of base pairs that are simply Watson-Crick base pairs. But in addition to these Watson-Crick base pairs, RNA can have a third type of base pair, so not just AT and GC in both directions, but also GU. So basically, the uracil, instead of a T, actually can base pair with a U in a slightly different way. Okay, so you have more base pairs than are possible in DNA. You have one more base pair. Yeah.
How does the RNA fold in the tertiary
there's a lot of additional atoms around. So basically, you can have these, this part interacting with that part, and the hydrogen bonds are sort of helpful in the middle because they're, you basically have, but you can have hydrogen bonds between more than just these hydrogens that are sitting there. You could actually have other hydrogen bonds. You can have both hydrogen bonds as well as other types of molecular interactions there. So basically, these molecules can actually share more than just these interactions. Does that help? And part of that is just simply biophysical properties of what are the stretching forces on the different base pairs as they're sort of looping around. So you basically have this phosphate backbone that's stretched in all kinds of ways as the two-dimensional form folds, which will then naturally lead the RNA into, first of all, its natural sort of kurtosis, if you wish, or its natural twisting, and other properties. Does that, yeah. Any other questions? But a lot of RNA folding research focuses on this two-dimensional structure. It's much harder still to infer the three-dimensional structure. We don't have the tools yet. So the secondary structure is very important because it covers most of the free energy of the folding. It is a biochemically useful coarse graining of the 3D structure, and it is actually a physical intermediate of the folding pathway on the way to the full 3D structure. And it is often very well conserved in evolution, and it's also computationally much easier to handle than the three-dimensional structure. I like to say that 2D folding is still computer science. Three-dimensional folding is more physics. It's like biophysical, property. Whereas here, it's still sort of information-based and, sort of easily algorithmically approachable. And there's many ways of representing this secondary structure. So if you look at the tRNA again, you basically have this cloverleaf structure that can be easily visualized in this plot that I showed you earlier, where you basically lay out every nucleotide this way. Or you could lay out all of the nucleotides in a circle and simply draw lines between the nucleotides that are connected. Is everybody with me on this one? Alternatively, you could basically build a dot plot. A dot plot is a scatter plot that basically has a dot for every base pair, effectively. So you draw the sequence here and there, and then you ask what are the self-similarity patterns of that sequence, and also what are the folds of that sequence. Okay. So for example, here you can basically see that the green one at the beginning, then the red one in the middle, and then the blue one here are in fact matching the green one at the beginning, the red one here, and the red one, the blue one here, and the red one there. So basically, if I start from the beginning to the end of my sequence, and again from the beginning to the end of my sequence, you can see that the beginning folds with the end, and then the middle with the middle, or actually I should say the first third with the second third, and then the second third with the first third. Okay, does that make sense? So basically, this representation captures exactly that. It basically tells you who folds with whom. If I take this out and I lay it out straight and this way, then I can actually represent this in a dot plot. Who's with me on the dot plot representation? Great. And then the last one is this mountain plot. That's basically, in computer science, there's push and pop machines. So basically you can push, 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 pop, 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 push, 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 pop, 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 and then pop, 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 pop. And that basically tells you that I'm starting, you know, a loop here, and I'm only going to close it there. And in the meantime, all of these open and close will happen here. So it's effectively a nested parenthesis kind of representation. And in fact, you can actually represent folding also in a nested parenthesis representation. You can have basically the exact sequence and simply open a parenthesis every single time there's a base pair, and then close it whenever that base pair is done. Okay? Who's comfortable with all these representations? Raise your hands. Awesome. Great. Yes.
Is there ever an instance where RNA would do like a half turn and bind parallel to itself?
Yeah. So basically, you're asking about, for example, this part here that could loop out to that part there. Is that what you're thinking? Or are you thinking about ways in which the order is not preserved? Yeah, yeah. That's a great question. No, there shouldn't be, because of the antiparallel nature. So basically, it's not a symmetric molecule, it's an antiparallel molecule. So 5' to 3' only binds 3' to 5'. So that's sort of the reason why you don't see that. It's a great question. Any other questions? All right, so these are the representations of it. So how do we actually figure out the structure? So again, the parenthesis representation is you know, the same one that we have here. So, and here's the double representation. So how do we actually measure that? So the simplest way is to effectively write out a dynamic programming algorithm for folding a sequence onto itself, okay? And that's where the very first algorithm for this mapping came from. And this is known as the Nussinov algorithm. So the idea is the following. I'm going to align a sequence from start to end and from start to end with itself. And the way that I'm going to do that is that I'm first going to compute all high similarity local alignments. So I can basically construct a local alignment in the same way that I could have constructed, you know, just an alignment between A and B. I'm going to create an alignment between A and then the reverse complement of A. And then these are going to be the high scoring local alignments. And what we want to find is a path that allows us to fold the molecule by utilizing the maximum number of these local alignments. Okay? Who's with me with this intuition? Perfect. So this would work fine and well, except for the fact that this is a single molecule onto itself, and we cannot sort of reuse these sequences because, you know, it will already be utilized for something else. So basically, if this part is aligned here, this part cannot then realign somewhere else because it's the same sequence. Okay, that's part one. And part two is that RNA folds have actually these bifurcations. You basically can have, you know, within this set of parentheses, I can start another one and close it. I can start another one and close it. I can start another one and close it. In other words, I can actually have this nested structure of I'm in the middle of a fold, and then I create another fold in the middle of that, and I create another fold in the middle of that. The easiest way to do that is to basically say, how can I create such a nested representation of folding? And this nested representation of folding is very easily done from the middle out, where you basically say, well, let me see what is the best fold between this part and that part. Well, it has to do with the best fold of any of the subparts. So I'm going to be basically breaking up this problem of folding into sub-problems using the exact same dynamic programming methodology that we learned about earlier, but with an additional condition. The same methodology applies when I extend by one nucleotide, or extend by a pair of nucleotides, or extend by a gap, for example. So, you know, this idea of sort of I'm including part of a fold with one offsetting nucleotide. And I don't have an example here, but there were examples there earlier. And this is basically done by saying that given my larger sequence, I can basically define this dynamic programming matrix that basically tells me what is the best optimal energy of folding a sequence from I all the way to J. Okay? And this could be done by expressing it as a function of the subproblems by including one nucleotide on both sides. So basically I'm extending, I have a loop now and I'm extending the loop on both sides, or I'm extending only one side with non-pairing on the other side, or I'm extending the other side with non-pairing here, or I'm including a whole other, one of these bifurcation events that I mentioned earlier, okay? So basically it's gonna be the maximum among 4 possible choices where I can actually do that bifurcation anywhere in the middle of my sequence. So I could basically, to fold I and J, I could extend this way, extend that way, extend both, or choose a midpoint and then recurse on this side and on that side. Raise your hands if you're with me. Awesome. So that's what the Nussinov algorithm does. It basically starts with the smallest folds, and these are the ones along the diagonal. And then it moves out from the diagonal, which are larger and larger subsets of that sequence. So basically, I'm aligning from the middle out and then creating larger and larger nested structures, okay? So basically, the recursion formula is that, for I and J pairs, it's either, including, one base pair here or one base pair there, or including the bifurcation event in the middle and then nesting here and there. And then I'm basically going to minimize each of these options. And then the traversal order is I'm moving from the diagonal out in pretty much any order. And then once I found the optimal score, I then backtrack and trace what were the series of maximum choices that I had to then arrive at that optimal solution. Okay? Everybody with me so far? So basically, when I arrive here, I then trace back as to what are the series of steps that gave me that optimal. All right, so that's the Nussinov algorithm. That's sort of the basic algorithm, and this is this basic dynamic programming algorithm. You can then go a step further and say, well, the most important aspect of RNA folding is not in the specific base pairs that are made, but it's in the stacks, so in the pairs of base pairs, and that's where most of the energy resides. So basically, this hydrogen bonding provides, yes, some energy, but the interactions between consecutive base pairs provide a lot more energy. So much more biophysically realistic folds can be made using these loop-based energy models. So instead of looking at a single base pair, you're basically looking at all these stacking interactions between neighboring pairs. Everybody with me? And there are many ways of calculating this. You can basically train this model by looking at what is the energy of stacked bases, what are the energy of bulges, of internal loops, of hairpins, of multi-loops, and stacked bases and hairpins, and then simply calculate all that as you go through this alignment matrix. And biophysicists basically calculated the stacking energies between every pair of paired dinucleotides. Okay, so UA, AU, CG, GC, and then also GU and UG. And you're basically saying, well, if I have a CG stacked with a GU, what is the stacking energy of that? Everybody with me? So using these stacking energies, you can now write a recursion that's based on these stacks. And you could basically have a fold be a fold with some additional loop here, or a hairpin with some interior looping, and this multifold, and so on and so forth. Okay, so you can basically write out these recursion rules that allow you to express these more complex objects as parts of simpler objects. So this is at the basis of it all, and then beyond that, you basically want to know what is the full probability space of pairing. You don't want to just say what is the best fold, but what is the space of folds? And one way to do that is to basically look at the full thermodynamic ensemble rather than this is the most likely structure. You can basically say what are all of the other substructures that might not even appear here. And you can basically see here that there's a lot of energy into base pairs that sort of fold here. And that is in fact competing with this one, for example. So you might be alternating between these two structures, and the structure of the RNA might be breathing between these two different forms. And it can basically tell you, how much of that energy is captured here uniquely versus how much is scattered between multiple intermediate places. Is everybody with me here? So this is all biophysical. This is basically saying, let's calculate a bunch of energy constants and then use these energy constants to infer what is the most likely fold. Another way to do this is to basically do this purely in a sort of more computer sciencey way using grammars. What you can basically say is, I can generate a parser for RNA structures that allow me to fold them into these nested structures. And the simplest model for that are context-free grammars, which were invented by Noam Chomsky, who actually used to have an office here just on the 8th floor of the D Tower. So a context-free grammar is basically a set of generative rules that basically say, I can explain any kind of, sentence as these constructions. It basically has a set of non-terminal symbols. These are the internal states. A set of terminal symbols. These are the characters that are being produced. And a set of production rules that basically allow me to transform these into those. And it has, a starting and an end, okay? So you basically start with V, which can be, any set of S, and then S itself can be, this, and so on and so forth. So using that, you can basically say, I can express any RNA structure using this very simple grammar, where my sentence can be either nothing, or it could be two neighboring adjacent structures, or it could be a single nucleotide to the left, or a single nucleotide to the right, or pairs of nucleotides, AU, UA, GC, CG, and so on and so forth. And now I can basically express my sequence by parsing it. I can fold my sequence by parsing it through these grammars, by effectively generating the A and the U together, and so on and so forth. So for example, I start with this. I mean, this is all the way to the bottom, but basically I start with S, and I use this rule that says S can be either of this. So I use the bifurcation rule. And then I emit AU and CG and AU and GC together as pairs. And then I emit a bunch of sequences on the left. And then I do the same thing on the other side, and then emit a bunch of sequences. So that basically parses this string here at the bottom into this folded structure. Who's with me so far? Awesome. So these rules simply tell you what could potentially be an RNA molecule fold, but it doesn't tell you with what probability. To look at what probability, what we're going to do is add a probabilistic component to every production rule. And that's how we make a stochastic context-free grammar. So a stochastic context-free grammar basically has the same thing, but it has probabilities associated with every rule. So you can basically say this has higher score, or than that, and sort of, or lower penalty or something like that, to basically generate sequences according to some probabilistic model. And these stochastic context-free grammars become even more interesting when you put them on a phylogenetic tree, and then they become philo-SCFGs, okay, stochastic context-free grammar. So these basically allow you to now express not just a single sequence, but the alignment of a sequence across multiple species using these production rules. And why is that helpful? Because if you look across species, there's a lot of conservation of structure, even though you may not conserve the primary sequence. So for example, a particular sequence might be replaced with another sequence that folds equally well. So in your stochastic context-free grammar, instead of generating a single sequence at a time, you generate a column of your alignment. And then you can generate paired columns of your alignment from the inside out in a parse tree that basically has scores associated with particular substitution patterns, okay? So, and then of course you can combine all that. You can combine biophysical information and phylogenetic information, and you can solve kinetics and pseudoknots and RNA interactions and so on and so forth. So basically the kinetics tell you what are the energy thresholds that have to be crossed in order for a sequence to fold, and what are the processes, the steps through which this fold happens. Pseudoknots, as I mentioned earlier, are connections that can happen across distant strands that basically break this nested parenthesis assumption. And these are much, much harder to fold. And you can also look at interactions where one folded RNA molecule might actually interact with another folded RNA molecule, okay? So the take-home message is that there's a huge functional spectrum of these RNAs, which is practically unlimited, and that RNA structure is critical for these functions and can be predicted computationally, and there's many different ways of doing that. And phylogenetic information can help improve the structure prediction, and this whole field is hugely active. There's still different classes of RNA functions that are being discovered and different classes of RNA molecules that are being discovered. And, you know, we all started with this ancient RNA world that eventually invented protein, and together they invented DNA. And then there's many ways of studying this RNA. The first is to map transcripts, either de novo or by mapping to the genome, and then to actually start folding these transcripts in 3 dimensions. Who feels that they learned stuff today? Yay. All right, see you guys tomorrow at 2 p.m. for office hours, 3 p.m. for recitation, and 4 p.m. for the mentoring session. They're all optional. You can choose to skip them. I would not recommend it. And then on Tuesday, we're talking about epigenomics, and next Thursday about 3D genomes.